Hello, everyone. Welcome to the Neural Implant Podcast. Today's guest is Thomas Stieglitz, and he is a professor at University of Freiburg in Germany, and he works on the reliability of neural interfaces. We've actually known each other for a while, but he finally, now with scheduling is able to come on the show. And we actually have a funny story. I didn't really mention this on the show, but I thought I figured I'd share it here because it's just so embarrassing that I have to do it. So I think it was 2018 or 2019, Society for Neuroscience. We're flying out, and I flew on Halloween, October 31st, and I'm like, okay, I've never flown on Halloween, so I'm gonna wear a costume. And so I have this giant chicken or giant duck costume. It's like a full-size onesie thing. And so I flew with it. Turns out nobody else was flying with a costume. Maybe there was some Starbucks baristas or something that had some like angel wings or whatever, but that or the halo. But otherwise, it was just me that was wearing a costume. So it kind of looked like a jackass. Anyways, on the flight over to San Diego, I saw Dr. Stieglitz, and I'm just like, oh, I have to say hi. And I can't imagine what he was thinking when I came up to him and started talking to him in a duck costume. But anyways, a few days later, I went up to him in a poster session, and he kind of looks at me and says, "You're wearing less yellow than you were before." So that probably tells a little bit about us and our interactions, and so hopefully you'll enjoy this show. It's a good one. It's an informative one. Thomas Stieglitz, pleasure to have you on the show. You are one of the guests that actually, it's kind of funny. Like, I've known for a long time, and I remember talking to you at Society for Neuroscience in 2017, almost 4 years ago.
Wow.
And just somehow, like I don't know, we're never able to come on the show, or I don't know, the timing was always wrong. It's kind of funny because some people I meet through the show, and then some people I meet 3.5 years after I've met them, or they're on the show. So pleasure to have you on the show finally. And then now it's a little bit different between us because I am now working in kind of the rival lab. I'm working in the Jack Judy lab at University of Florida. And so now we're kind of the ones that are like, working on the same stuff, obviously still respectful and good and everything like this. But so it's kind of interesting now. But pleasure to have you on the show. How are you doing now during this COVID time?
Yeah, pleasure is on my side. Things are going a bit slower than before, which is not always bad because you can really finish some things there. And to come back to the rivalry, I think that's really exciting. When I started ages ago, being, let's say, more or less the only one, then it's a bit lonely, and you never know if you are running in the right directions or not. So if there are others who running with you in the same direction, then it feels at least better that you know probably it's not that stupid what you're doing there.
Yeah, you don't feel like a crazy person, right? And so you talk about reliability of neural interfaces, neural electrodes. Why is this important? And actually, how long have you been doing this? I think since at least 20 years, right?
Yeah, 25, 26, something like that years. I think it depends what you like to do. If you want to be the, let's say, prototyping person, being the first one ever doing things, then it's not so much about reliability. Then it's inventing new devices, discovery of new mechanisms, whatever. But my driving force was always on the translational side. That means bringing ideas into patients. And that means you should start between the idea and the patient with some chronic preclinical testing to prove reliability. And reliability means that things do not fall apart at one point, that if you do it twice, you get the same results. Sounds stupid, but that's very important. And that it's not only about the basic mechanism, but that you are able to put cables on connectors on that you can clean it, that you can sterilize it, um, and that you have a yield that is probably better than 20% or something like that. That's all things where business persons would say, what is the guy talking about? That's kind of common sense or prerequisite if we're talking about cardiac pacemakers or cochlear implants or deep brain stimulators. That is the prerequisite, and that's taken for granted for each and everybody who buys an approved device. But in research, we are not talking about approved devices, but trying to use whatever technology that has been developed for something different to transfer that into neurotechnology and make new things happen in neuroscience. And I mean, when probably I started early on, but I can imagine when Ken Weiss started 1969 to say, hey, in Silicon Valley, they have managed to integrate 16 transistors to make the first integrated chip. Let's do some different things and stick that microcontroller into the brain. That people were saying, this guy is so crazy. Close the door and lock it and don't let him out. And it took 30 years or something like that before people say, hey, that was really cool idea to take that silicon and combine brain probes with microelectronics. And then it was another long way, if we look on the Utah array, really to say, well, this is a piece of sand, and I make a 100 channel or 96 channel array out of that, and I shoot it with an air gun into the brain. And by the way, that is a human brain, and we still have a socket that pokes through the skin, and we put something on top of that. That sounds crazy even today, but this is now an IDE for human application. And I mean, this is something where it really takes a long time to test stepwise. Does it really stick? And when I came up with that idea of polymers, or I did not invent that, but I stole it from others and then worked so long that I could show, hey, it works somehow in on the brain. You can put it in for a year or two. You can put it in humans for 6 months, and the yield is still better than 80% at the very end. This is a small step, but I think it's an important step towards transferring more things also now in bioelectronics medicine, into applications where it's not just making a rat walking in a maze, but really to helping persons to regain quality of life or getting a better treatment.
Yeah, definitely. I mean, based on what you're saying too, it really sounds like, research is, it's based on PhD students who are around for 3, 4, 5 years. So they don't want to be doing an experiment for 1 or 2 years. So usually their experiments are very short. So they don't really care about the longevity and kind of durability of these devices. So yeah, it's not until you start to transfer that into like a inhuman model where it starts to matter. So yeah, I mean, I always kind of thought of you as the, I don't know, yeah, the polymer guy, the guy that brought polyimide to the field. So what is the benefit of polymers over sand, as you said?
Well, I think one advantage is that they are more flexible than silicon. They are not as flexible still as the brain tissue is, but they are more flexible, and you have a larger variety in shape. And something that persons from micromachining not say that often is you can make larger devices out of that. One thing is to make things really small, but if you make things really small, you have to know where to put them, right? If you can target a single nerve cell, you must know that the nerve cell you're going to target is the correct one. While if you have something that's larger, you can include a kind of redundancy. You can implant that, and then you can check afterwards which is the best contact that I could use. And if this changes over time, you can switch in between contacts, which is good on a shank like a Michigan probe, where it can go from bottom to top. But it's hard to go 1 millimeter to the left or to the right if you just have one shank with a linear array. And I think this is not bashing now silicon technology. Over the years, it came clearly out there are applications in fundamental neuroscience where silicon technology is superior, and it's the best technology possible to record and stimulate. And there are other approaches where say, well, with that idea of having local field potentials or networks of networks that I want to investigate, probably it's more clever to have something that covers larger areas, and this is where polymers come in. I mean, the last example of excellent public relations and marketing was Elon Musk Neuralink. That is also polymer because it's so soft and flexible. And I mean, there these terms of soft and flexible are more colloquial. If you really go into the into the mechanics and the engineering part, you still see that there is a mismatch of orders of magnitude. But I mean, if it works well, you can proceed. And this is what I found out in my experience. Um, Polyimide doesn't harm the tissue that much if you design it well, and it's still surgeon proof. So it's tough enough that a surgeon with not bashing surgeons, but they have a certain training, and surgeons can do things that engineers can't do, and the other way around, right? But in a general surgical procedure, I think polyimide performs quite well, while Parylene C, for example, in the same thickness, in the same width, so same design, is probably a bit too, well, not tough enough. That means you might destroy it before it gets implanted, which cannot be the ultimate goal, right? So you have to find that balance between softness and usability.
Yeah, definitely. Polyimide is very, very robust, yet obviously softer than silicon or glass. So, but it's actually interesting too because so obviously you put something in the body doesn't like it, the body tries to reject it or encapsulate it, push it out, whatever. But kind of what you're seeing a little bit too is like the polyimide might be even better than the silicon in terms of the biocompatibility or that there's that it's even. I mean, people think of silicon or glass as very inert, right? But this is even more inert than that, right?
Yeah, that's. I mean, one has to try it out. It depends. If you do everything correctly or ideally, then silicon is really stable. If you have boundary conditions like temperature or certain process restrictions, then you create layers that the body eats up. Um, since the substrate itself is relatively stiff, the metallization on top of it sticks quite well. This has been a challenge with all the flexible stuff. If you have metals and then you pull it or you bend it, you get a lot of interfacial stress there, and the metal tends to flake off. So it was a hard way to find out how to make it stick, and we're now pretty confident. There are other groups having different approaches there. That's all fine, but I think that integrity and stability is an issue for sure. And
Okay.
also the question, what is that good for? If you really want to go into fundamental neuroscience and animals for half a day or a week, or is our goal a transfer into humans and 10 years or 50 years or something like that?
Okay. And so you've also done a lot of work with PDOT, polyethylene. I got to look this up.
You have to say it in long. I'm not able to do that without reading it.
polyethylene dioxythiopene, and then the PSS is usually the
Yeah.
most popular one, polystyrene sulfonate. So, what is this material? And what's so good about it? I actually we talked to Maria Asplund of TU Freiburg a few years ago back on the show, but
So
you guys worked together,
she
this right?
might have told you all the truth about that. So she knows much more than I do about PDOT. It's a conjugated polymer, and it's a proton conductor. Or let's say an ionic conductor, to generalize it more, and that's beautiful because all the salt water in our body is conducting by ions, and all the electronics part of the technical stuff is mainly conducting by electrons, and then you have all the challenges where electrons have to be transferred into ions, this is where the metal electrode meets the salt water, and that would then cause some electrochemical reactions that are sometimes either detrimental for the cells or destroy the electrode if you don't take care. And the smaller you are, the less surface you have, and the less surface you have, the less interaction you can get. And that will mean you screw up your properties either with respect to recording. So impedance goes up, noise goes up, and the signal to noise ratio drops. That means you can hardly discover your nerve signal and all that wiggle waggle of your recording. If you would stimulate, you don't get much charge over the electrode, and if you push harder, you destroy the electrode. To really get it short and simple. If you put PDOT on top, if it's not just the surface, it's the volume that you have. And the volume, even if it's just a micron or something that is negligible, is so much more than you get with a metal electrode interaction, and that means you take benefit of those surface effects, and thereby you reduce the interfacial impedance, you increase the signal to noise ratio, and you're able to push more current through the electrodes. On top of that, it's softer than polyimide or metals. That means you have a kind of something soft towards the tissue. A second advantage, and the third is if you really know what to do, like Maria does, you can load that with drugs instead of PSS, and with that you can drive regimes where you can either record and stimulate, or you can drive out drugs that actively tailor some of the reactions in the vicinity of the electrodes.
Yeah, it's a very exciting material, and it seems like you guys are working on all the exciting materials involved in neural implants. So yeah, I mean, if you could give us like a cheat sheet or just a brief overview, what's the best, most reliable neural implant? Like, what do you have to do? Like, what are the ingredients for that?
I should wear now a pink coat and pretend to be a fairy. Well, the ideal implant should be probably of no size, invisible to the body, and I can stop here. If you have no implant, that would be the ideal implant. Um, what is of importance that you know your target very well. So it means to tailor the functionality to the needs. So general purpose implant would not work. So at least to my impression, so it must be really tailored. Should be as robust as possible. And saying that, I would say if you can afford using old style technology, like titanium cans, thick silicone rubber housings, um, MP35 helically wound wires, really like a cardiac pacemaker, something like this. Go for that. that's robust, that's reliable, and it's a kind of predictable what will happen. You can estimate the lifetime. You can take advantage of all the experience of 60 years of cardiac pacemaker technologies. Um, However, if you need much higher complexity, let's say 100 channels in the brain, just to be sure that you cover everything, that you're able to follow the signals when learning takes place and the signals shift probably to one side or the other or whatever. Um, then you should not only consider the interface, but also the whole concept. And I strongly believe that the secret for reliable neural implants is the assembling technology. So it's not only about the interface, it's not only about the electronics, it's not only about the package itself, but it's really how do I connect one piece to the other? Do I understand what the materials will do? Have I really considered failure modes? And this is one of the white spots on the map of neural implants. So we are talking about the 21st century as a digital age, but I think what I would love to see, not necessarily with my name on that paper, is a really comprehensive model of a digital twin of implant failure modes. Yeah, that you have a kind of Lego building blocks and can put things together and you know how it works. And this is a great dream. I know that it will not work as I would like to have it because uh, boundary conditions in the spinal cord are different to the peripheral nerve, are different to the ear, are different to the brain. But to be able to transfer building blocks from one piece to the other and take advantage of the knowledge, that would be really great.
So you're saying based on experimental data or computational data? Is that what you're saying? Or
Both. So my feeling is that the failure mode models are not yet comprehensive. Yeah. We all know if you put silicone rubber on something else as encapsulant, it should stick. And depending how you clean the substrate and what you do and which silicone rubber you use and how you spin coat or cast it, whatever, it will fail sooner or later. But what parameters do I have to put into the model to see if I have this material, this substrate, this casting technology, and that size, and that thickness? So what are the parameters that influence my failure, and could I use, let's say, whatever software package first to simulate the design and say, well, this might fail here. The encapsulation will fail after 20 years, and the feed-throughs will fail after 25, which is fine because first, yeah, it's the right order of failure modes. And and I say, well, this is something that will be implanted into patients with a remaining life expectancy of 15 years. We have a safety margin of 10 years. We can go. And then
wow,
doing
that
a
would be
reduced number of animal experiments and whatever and transfer it into humans. So that would be a dream.
that would be amazing. That would be, but then I mean that's the question too, is like how do you test that? Because if you say it's going to last for 25 years, then you're going to have to wait for 25 years to get the
Yeah,
real
definitely
results.
not. I mean, that's all those accelerated aging models and those multi-parameter things. This is something, it's difficult, I mean, in the pioneering days, when we look on those historical things like the cardiac pacemakers, where people, well, at least it is said that people took an empty case of tin can of shoe shine and put their 5 to 10 components and a lot of epoxy in there, and then it got implanted. Um, Now we have to do all the tests, and of course not for 25 years, but at least for 5 years or 6 years or whatever, before you're allowed to go the next step. And This is very important on one side, but I think that safety expectation on that side um, is really blocking a lot of persons from switching on their entrepreneurial gene and bringing something into a product. And it needs persons with a amount of money like Elon Musk and his vision, and I think he's a really pushy person there, bringing those persons together and having probably in mind if it doesn't work, I just burned 150 million US dollars. Yeah, this is not everybody wants to have such a career goal, and then it's really hard to get venture capital on a timeline of usual VC offices of 2 to 5 years. To have something like that. So um, that's, I guess, one of the larger challenges in that field if you really want to bring something to the market.
Yeah, definitely. I mean, it's a big risk. I mean, obviously you could be endangering people's lives. And if you have a bad, the product recall on something like that would just be potentially really bad. And that's why we're limited by, deep brain stimulators, which are essentially pacemakers, which have been around
Yeah.
for 50 years or whatever. And there's not really too much, advancement because people are scared, and these,
Yeah,
these
people are
big,
scared. They're not scared drinking 2 liters of Coke and eating two burgers, but they're scared of 5 grams of silicone rubber. You know that.
Although, 5 grams of silicon rubber inside your brain, like that's kind of, I understand being scared
Yeah,
about
yeah, I
that.
completely understand it. I would not. I'm really one of those who try to do those developments really really responsibly. Not promising too much and not bringing things that are not mature enough into humans. But I think at one particular point, would be nice to have the opportunity that you, for example, have in the US with the IDE to start with at an early stage and a really restricted framework to bring something that is not a fully approved product into human clinical trials. This is, for example, something that's completely missing, that idea of that concept that's completely missing in the European legislation. So we have nothing like an IDE. So you have to negotiate in each and every clinical trial with the legal entity of your particular European country what they want to have, how you have to prove that, and how to succeed.
Yeah, yeah, definitely. I mean, I think Americans are generally more risky, especially Germans. Germans are like, very much like, okay, it should be perfect, I guess, and
Yeah,
optimized and everything like this. So, but
perfect.
but I mean, you have experience with this because of the company that you I don't know are on the board or helped found. I mean, obviously you weren't like the main founder, but Martin Schuttler with Cortec. Do you want to talk about this a little bit?
Yeah, this is something where we have experienced a lot in changing legislation and stuff like that. And Cortec has now an FDA approval for the implantable epicortical grids. Um,
Congratulations.
and they are working on a fully implantable system, let's say, as a kind of research platform for bidirectional closed loop applications, mainly in the brain, with the idea that you normally do not know when approaching new therapeutic ideas what you really need. Of course, it must be safe, it should be reliable. That's not the question, but you do not know, do I need 4 channels? Do I need 8 channels? What signals do I need? How can I feed them back? What is a good algorithm? And the idea behind that brain interchange technology is to have a kind of platform system where you can do clinical studies in humans on a safe level to exactly derive those data to be able either use that system or by knowing the data and knowing the disease to say, well, probably we should tailor it a bit into that direction and can use it in a more efficient manner than if we would, let's say, cut off those 8 channels and focus on that particular parameter. Now that's a really long and rocky and winding road to go stepwise to the next level. I think they're on a good way. It's took longer than expected, but I think they're now really on a good way and happy to see the next things there.
Yeah, it must be exciting. I mean, basically implementing the your research, into the market, I guess, and slowly. I mean, it always takes longer than expected, but yeah, no, I think that's really. So, how much of a role do you play in that? I mean, you're just kind of an advisor for it, or
I'm
what?
just the old guy in the background. I'm a consultant in the scientific advisory board. That means I have no active role at all, and that's probably good because my job as professor already is already more than 100% 9 to 5 job. And I think it really takes persons who invest their more than 100% of a 9 to 5 job in that company. And therefore, I'm more like scouting what is out there and giving advice from a certain distance to that.
Okay, yeah, that's understandable. Yeah, very cool. So you also have some papers about explants and basically comparing or looking at devices that were removed from the body. So what does that look like? What is, I don't know, the
I mean,
damage to the devices?
it's always for me. It's very important not only to share the pleasant things, but also the dirty ones. Um, and we have had some amazing implantation studies um, where the first patient performed extremely well for 30 days. That's the limit for the first in human trial. And then things got explant. We say, hey, give them back to us. We want to like to have a look on that. And the patient performed extremely well up to the last day of those 30 days. And then we got only broken pieces back to us. Hey, how can that be? If it works in the human body and you put it out, and we just get bits and pieces. And then we understood, well, we had to pull a little bit harder, and we want to save the nerve and not the. And so, okay, part of that work might be probably because persons did not take care to get it out smoothly, but just scratch over it. But it's if you have only bits and pieces and not a complete thing, you don't know. And if you ask the surgeon, do you agree that you destroyed that? Of course, nobody would say, yeah, of course, I destroyed everything. But they will say, well, we don't know. But we found out that adhesion was really lousy. And then we thought, well, that's not good going to chronic studies when we know things fail. And we put in those adhesion layers. And then 2 implants failed after 2.5, 3 months. And it turned out they failed because some of the rehabilitation personnel was not willing to follow the user's manual, and they broke off the connectors. That is the
Hmm.
worst thing that could happen, right? That the internal part might work, but somebody screwed up the external part. And we were, for some stupid reasons, not considering that. We improved that in the next round, and we had yield of more than 80%. And then we took all those things that we could get, documenting what worked electrically before explantation and what did we see after explantation with those bits and pieces, and tried to make an arrangement of that. And we still have some unknown unknowns there. Um, but most of the damage was so serious that you could really see how the pair of tweezers poked through from the backside or something like this. And I mean, then it's really fair to sort that out as mechanical failure of explantation. And we believe now that we can predict with the data that we have and some additional in vitro experiments that we did after getting them back to proving something, that those pieces can survive up to 5 billion of stimulation pulses. which is, according to the last experiences that we have, probably a lifetime of about 6 years um, in daily use when you have sensory feedback of a hand prosthesis or leg prosthesis over the day. And I mean that I think that's pretty fair to continue with a performance like that, um, either for other applications or for some other studies. We still the major challenge now is and that's the same challenge that Dustin Tyler from Case Western is facing, to integrate that stuff now into a fully implantable system. So we all started with percutaneous wires. Max Otis Catalan has that beautiful solution that they're using metal rods in the bones transcutaneously as anchoring devices for their prosthesis, and they funnel the wires through those sockets out, so they don't need no wireless connection through the skin. While Destin Tyler and our group with Sylvester Michera, um, we want to have something wireless, and this is still a challenge to find out how to connect those many channels to an implant electronics. And the for persons being not in the middle of that field, it's not about cables, it's not about packages, it's really about the connectors. How to connect that stuff in a way that you could exchange a single piece. You don't want to solder four cables with electrodes at a tip permanently to a tin can or to a titanium can. That would look like a jellyfish or like an octopus or whatever you can imagine, an animal like that, and how to implant that, and what do you do if you screw up one of those arms during implantation, right? It would be nice to have different pieces that you can implant, and if you screw up one thing, you can exchange it, you can connect it afterwards, and have it more modular. And this still a challenge in the field. That's even less sexy than reliability with respect to convincing PhD students to work on a topic like that.
A connector, something that connects to this, and it's reliable. Yeah, that's actually one of our projects is the connector project, and so yeah, it's not sexy, but essentially making a plug, that you plug into, but. Yeah, I mean, I think it's limiting the field because, right now they have the ball seal connector, which is what they use in, deep brain stimulators and, that kind of stuff. But that's limited. It only has like 6 channels or 8 channels or some 16 channels. And so if we want to get to 1,000, how do you do that? Right. And so
Right.
that's the dream is 1,000, 10,000, 100,000. And that's a lot of soldering, 100,000 soldering joints. That's a lot. Yeah, so it's difficult, but it seems like we're very much on the going on the same path and doing that, learning from each other. I think too.
Mhm.
But yeah, well, Dr. Stieglitz, this has been excellent. I've really enjoyed this. I kind of want to. I mean, I know that we can get deep into the weeds and that we can have a much more involved conversation. Talking about the details and everything like this. But kind of want to keep it a little bit more surface level for the show. But yeah, it's been a pleasure having you on. And is there anything that we didn't talk about that you wanted to mention?
No, I think we have covered all of, well, not all, but a lot of exciting things. It's always the challenge to keep the balance between the width and the depth of that topic.
Yeah,
I think that's
yeah,
fine
for
from,
sure.
from my side.
Excellent. Well,
Again, was,
thank you so
was
much.
as usual a pleasure to talk to you.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.